In a previous video, I described something called the central dogma of molecular biology. Dogma, that is a very strange word for scientists to use. Dogma is something that is, it's like a written law. Usually you see it in religious contexts. So for something to be called the central dogma of molecular biology to be used in science, that tells you it's a pretty darn important idea. And here's what it is. We've known for a while that proteins do everything interesting inside your cell. They do all of the work inside your cells, I suppose I should say. They are the enzymes, they're your contractile proteins, let you move your muscles. They are structural proteins, collagen, they hold your tissues together. They allow things in and out of the cell. Proteins are what actually gets life to live, so to speak. However, proteins cannot code for themselves. They do not have the instructions for making themselves worked into them. So in order to make proteins, we need an information source, and that information source is DNA. The nucleus of a cell, I like to think of, I've mentioned it before probably, I like to think of it as a central archive, a kind of big library with blueprints all over the place. Now those blueprints, you're not allowed to remove them from the nucleus. You're not allowed to remove them from the library, but you are allowed to make photocopies. You're allowed to copy that information into a portable form. So, what is that portable form inside the cell? It's RNA, ribonucleic acid. A molecule called RNA polymerase can come into the DNA, find one particular gene, right? A gene is just a section of DNA that codes for one protein, and it can copy the genetic information in that gene, and then that RNA molecule that's been copied is allowed to exit. If you've ever watched a movie where there's a courtroom scene, there's always someone typing away everything that someone's saying inside the courtroom.
When you say photocopying machine, what do you mean?
The document that she's creating is called the transcript. The transcript, it's a copy of what everyone said inside the courtroom. So this process of copying DNA into an RNA package is referred to as transcription. Okay, so you transcribe DNA into RNA. Now we give that RNA a special name. It's called mRNA, as in messenger RNA. It carries a chemical message from the nucleus to the ribosome. And ribosomes, we know about ribosomes, don't we? Ribosomes are what actually make polypeptides, right? They take genetic information and they synthesize proteins. You remember that from our tour of the cell. So the RNA, the mRNA, comes to these ribosomes and it shows the ribosomes the genetic information. And the ribosomes have to have a way to read that information and synthesize together the correct polypeptide. Now at that point, you're going from nucleic acid language over to protein language. So we need molecular interpreters. We need a way to translate that information. And the process of making polypeptides in the ribosomes is called translation. So this is the central dogma of molecular biology. In order to go from the information held in DNA to the functions conducted by proteins, you need to go through an intermediary. First, you need to transcribe the information into RNA, then you need to translate the information into polypeptides. Every chromosome is a single long string of DNA, and that long string of DNA can contain hundreds or 1,000 genes on each one. We have lots and lots of genes inside of our genome. The genome is all of your genetic material, all the chromosomes added together. For instance, the gene for making insulin is found on the 11th chromosome in the human genome. But the entire chromosome is not dedicated to making insulin. Only one little segment of that chromosome has the correct gene for making the protein hormone insulin, right? So you can see it's right there at the very tip of the 11th chromosome. That has the genetic information for making that protein in green and pink here in the upper left-hand side of the slide. So every chromosome has a bunch of these genes in its structure. But when we're making a protein, we're only interested in one gene at a time. So in this chart, you can see that the sequence of bases in the DNA, you get an RNA molecule that's base pairing in a complementary fashion to that one gene, to that one segment of DNA. And then for every 3 RNA bases in the mRNA, you get 1 amino acid. And this solves a problem that we mentioned in an earlier video. Early biologists didn't think that DNA and RNA were complicated enough to code for the rich diversity of life that we see out in the world. But what we realized is that if you have combinations of these nucleotides, you can have a lot more nuance in that genetic code. So for every group of 3 letters in an RNA molecule, you code for one particular amino acid during the process of translation. And we'll come to that idea a little bit later. But on this slide by itself, you can see that if an RNA molecule is 300 nucleotides long, it's going to code for a polypeptide that is 100 amino acids long, because every 3 nucleotides tells you about one amino acid. This flow of genetic information is related to the genetics section we're going to talk about in a bit. So here's 2 words that are going to come up in genetics when we are discussing that. I'll bring them up again at the very beginning of the genetics material. But you have an organism's genotype and its phenotype. Its genotype is talking about its DNA. It's talking about what genes are actually present. Do you have the dominant form of this gene or the recessive? Which version of a gene you have depends on what the order of nucleotides are on your particular segment of DNA for that gene. The phenotype is the result of those genes. If you change the genetic information, if you change the genes, the genotype of an organism, you also change what traits they have. So if you alter some DNA, then that organism's not going to make a certain enzyme correctly anymore, and they might end up being lactose intolerant, for instance. Okay? So your genes affect the traits that you have. And as you go from a single-celled fertilized zygote up to a multicellular organism, the function of your different proteins are what really control how you develop. They control how big your nose is going to be, or how square your jawline is going to be. They control the color of your hair by controlling what kind of pigments are produced. So your genotype is what we're talking about kind of here, what genes you have, what does your DNA actually look like, and then the result of the DNA is your phenotype. Again, those are going to be important concepts in the genetics section, but you can see how they relate to this information here. The 2 big processes we're going to talk about next are transcription and translation. Now, again, transcription is copying DNA information over to an mRNA molecule. Okay, so we go from DNA to RNA. We make a copy of it. We make a transcript of it. Translation is when we go from the RNA molecule to the finished polypeptide at the end. So let's talk about how that process works in more detail. Transcription is accomplished by another kind of enzyme, another kind of polymerase called RNA polymerase. So DNA polymerase 3 and DNA polymerase 1 were used in DNA replication, but RNA polymerase is used in transcription. Enzymes are named after what they do, as I've mentioned before. That suffix "ase" at the end just tells you that it's an enzyme. What kind of enzyme? Well, it's a polymerase, so it's an enzyme that makes polymers. What kind of polymers? It's RNA polymerase, so this is an enzyme that makes RNA polymers here, okay? So that's what RNA polymerase is. Superficially, this kind of resembles DNA replication. You have an enzyme that is connecting itself to the DNA strand, and then it cruises down that DNA strand, creating its own nucleotide on the other side. It's using RNA bases, which complementarily base pair to the template strand of DNA in order to create a little RNA duplicate of the information that's in that gene. But of course, it's not completely identical to DNA replication because we're making an RNA molecule and they do have some differences between them. So RNA polymerase is the enzyme that completes transcription. And here's what transcription would look like. We have 2 DNA strands in the double helix which are locked together, but only one of those strands actually has the gene that we're interested in transcribing. The other one has kind of a mirror image of it. It's for duplicating the information. Having 2 strands makes DNA replication very, very easy, but we don't need the information on both strands. We only need the information on what's called the template strand. The strand opposite to the template strand is referred to as the coding strand. So, just a quick review of the base pairing rules. If this is my template strand, TAC, CGT, ATT. Then my coding strand, which is made of DNA, DNA would have A, T, G. A to T, A to T, C to G. G, C, A, C to G, G to C, T to A, followed by T, A, A. Okay, so simple base pairing rules, and you can tell that these two strands are DNA because there are T's here. Now when RNA polymerase comes in and it separates these two strands, it's only interested in the template strand. So that is going to be what it uses to base pair with in order to figure out how to put together the mRNA molecule. So RNA polymerase is going to come in and it sees a T, well, T binds to A. So it's going to put an A there first. Then it sees an A. Now, if this was DNA, we would put a T, but there are no T's in RNA, so there we put a U instead. mRNA has A, U, C, G, and wherever you would put a T, you put a U instead. Okay, C to G, that never changes. C to G, G to C, T to A. Make sure you practice a few of these because people get messed up when they see a T. They just put a U for all of these, but you should only be putting U's across from A's on your RNA molecule. A to U, T to A, T to A. So this would be what our mRNA molecule looks like. Now, if you notice something, ATG, AUG, GCA, GCA, TAA, UAA. The coding strand looks superficially like the mRNA molecule, with the exception of the fact that all the T's have been switched over to U's. So when you're doing problems with this, when you're trying to answer questions about what the mRNA molecule and the coding strand would look like, they should look similar to each other at the end. I would suggest you do both of them individually, that you base pair rules with this template strand for both the DNA and the mRNA, and then double check at the end to make sure that they are consistent with each other, because if they're not, you may have made an error somewhere along the way. Now, each of these groups of three letters, AUG, GCA, and UAA, are what we call a codon. And each combination, each triplet, is going to tell the ribosome one particular amino acid to put into the polypeptide chain. But we haven't quite come to that yet.